Hello students, in this lecture we will be discuss about the remaining micronutrients. In the previous two lecture I have covered the macronutrients as well as two micronutrients. So let's start. Third micronutrient is the copper, and copper is a necessary cofactor for oxidative enzymes. In well aerated soil, you have to know that in well aerated soil copper is available to the plant as divalent in cupric ion form, and this cupric ion form a chelate with the humic acid in the fractions in the organic fractions of soil and involved in providing copper to the surface of the root as compared to wet soil where the little oxygen is present. Then this cupric ion form is reduced to the cuprous form, and this cuprous form is unstable and plant cannot uptake this. Next, the functions of copper primarily as a cofactor for a variety of oxidative enzyme. For example, copper function in the electron transport chain in photosynthesis as a carrier of, as we know that in etc, in photosynthesis the plastocyanin is come. So this plastocyanin function with the help of copper. Also the cytochrome oxidase, ascorbic acid oxidase also function with the help of copper as a cofactor ion. Next, as we see that the when we cut the apple or potato, then the surface is turning brown, and this brown color of the cut apple and potato is due to the copper containing compound named as polyphenol oxidase. Superoxide dismutase, which detoxify the superoxide radicals, is an important copper coenzyme. So this SOD function as a free radical scavenger, means it remove the all free radicals, and as we know that the free radicals are very harmful to the plant, uh, and they are formed by the radiations. So if they are not removed from the plant body, then these free radicals continuously damage the plant tissue and harm the plant. Last one is the copper deficiency disorder. This copper deficiency disorder including plant stunt growth, distortions of the young leaf, and particularly in the citrus plant when the young leaf become folded or they become die, and this disease is known as summer dieback. So these are the some disorders or deficiency symptoms occur due to the copper. And this is the picture of the peach plant where the young leaf are completely distorted or they become dead. Next micronutrient is the zinc. So zinc is activator of numerous enzyme and absorbed by the plant in a divalent cation Zn2 plus. It is activator of large number of enzyme including alcohol dehydrogenase, carbonic anhydrase, and help the copper SOD to which detoxify the free radical oxygens. Deficiency of zinc reflect in the disturbance of the metabolism of oxygen, means if the plant has deficiency of zinc, at that time the oxygen metabolism is disturbed, and oxygen is a plant hormone and found in the plant in the form of indole 3 acetic acid. Next, zinc deficiency in the plant have shorter internode and smaller leaf, and the disorder is caused by the zinc deficiency is known as little leaf, mainly seen in the fruit plants. Precise role of zinc in the oxygen metabolism is remain obscure, means it is not clearly known, but the level of oxygen in the zinc deficient plant is declined, and before the obvious symptoms of the zinc deficiency appear. Additionally, if we give the plant to the enough zinc supply, then there is a rapid increase in the hormone level and plant growth is resumed. Also, many evidence support that the zinc is required for the synthesis of the precursor of oxygen, means tryptophan. So tryptophan is a oxygen precursor. In some, according to some studies, Zinc is required for the formation of this tryptophan. Next micronutrient is the manganese. So manganese is an enzyme cofactor as well as a part of oxygen evolving complex in the chloroplast. So manganese is absorbed by the plant and transported within the plant again as a divalent cation like zinc. Manganese is required as a cofactor in large number of enzymes including decarboxylase enzyme, dehydrogenase enzyme, and play a critical role in the respiratory carbon cycle. Best known, the most studied function of the manganese in the photosynthetic oxygen evolving complex. In the form of magnoprotein, manganese is a part of oxygen evolving complex associated with the photosystem 2, where it accumulates the charges during the oxidation of water. As we know that during the photosynthesis, uh, this photosystem 2 continuously absorb the sunlight, and with the help of this sunlight, the energy is transferred to the oxygen evolving complex, and this complex break down the, or we can say oxidize the water molecule and release the free oxygen and electrons and hydrogen ions. Next, manganese deficiency is widespread spread in some area depending on the soil conditions, weather, and crop species. So the deficiency, mainly deficiency is shown where the pH level is low, means in the acidic soil, and high organic content. Manganese deficiency responsible for the gray speck disease in the cereals, and the disorder is characterized by the presence of greenish gray oval shaped spot on the basal region of the young leaves as shown in this picture. So these are the gray speck disease on the oat leaf where you can clearly see these are these gray spots. Next, it may cause the extreme chlorosis between the leaf veins as well as decolorations and deformities in the legume seeds. So in legume plants, it also causes chlorosis, means deficiency of manganese causes chlorosis and a decoloration as well as deformation in the legume seeds. So in legume plant it also cause some kind of chlorosis. Next one is the next micronutrient is the molybdenum. So molybdenum is the key component of nitrogen metabolism. The main role of molybdenum mainly in the nitrogen metabolism. Apart from that, this micronutrients required by the plant very less. Means it is the micronutrient which are amongst the lowest of all known micronutrients and primarily related to the role of nitrogen metabolism. Means apart from nitrogen metabolism, this micronutrients do not do anything for the plants. Although molybdenum is the metal, but its property more closely resemble to the non-metals. Means it is a metallic compound. In aqueous solution, it occur mainly as a molybdate ions, means MoO2 4 negative, and there are several enzymes required for the required molybdenum. For example, dehydrogenase enzyme and nitrogenase reductase enzyme. Both enzymes are required in the nitrogen metabolism. And molybdenum requirement of the plant thus depend upon the some extent of the mode of nitrogen supply. Means if nitrogen supply is means if the molybdenum is absent, then these two enzymes do not function properly. So dinitrogenase is an enzyme used by the prokaryotes, including those bacteria or prokaryotes having symbiotic association with the higher plants. They reduce the atmospheric nitrogen. So the main functions of this enzyme is that these bacteria use this enzyme dinitrogenase, and this enzyme convert the atmospheric nitrogen to the ammonia. Next enzyme is the nitrate reductase, found in the roots and leaf, where it catalyzes the reductions of nitrate to the nitrite and this conversion is a necessary step in the incorporations of nitrogen to the amino acids. So this enzyme convert the nitrate, so nitrate like NO3 negative to NO2 negative. So this one is the nitrate, this one is the nitrate NO3 negative, and it converted into the nitrite, this is the NO2 negative, with the help of this enzyme nitrate reductase. In the legumes, as we know that legumes have root nodules and they have ability to fix the nitrogen. So molybdenum deficiency give rise the symptoms nitrogen deficiency. Means in legumes, if molybdenum is absent or deficient at that time, both these two enzyme nit- uh, 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 these two enzymes nitrate reductase and dinitrogenase both are not functioning. When the nitrogen supply is adequate, a deficiency of molybdenum show classical disorder known as whiptail, in which young leaves are twisted and deformed. So this disease is appeared when the deficiency of molybdenum are occurred, but nitrogen supply are adequate. Means plant did not require nitrogen, but plant having plant having deficiency of molybdenum. So this is a classical example of a classical disorder of molybdenum deficiency. In this whiptail disease, the young leaf are twisted and deformed as shown in this picture. So these are the some young leaves, they are continuously deformed and twisted. So this is the normal condition, and after that, due to the continuous deficiency of the molybdenum, the leaf are continuously twisted and deformed. So this disease is known as whiptail disease. In some plant may exhibit the interveinal chlorosis and necrosis along the veins of older leaves. So this is another symptoms, another deficiency symptoms of molybdenum. Main deficiency symptoms of the molybdenum shown in the legumes as well as the member of Brassicaceae and maize plant, because they need molybdenum for the functioning of nitrogen metabolism enzymes. Next, molybdenum deficiency is worsen in the acidic soil with the high content of iron precipitate, which is strongly absorbed the molybdenum ions. So this deficiency is worsen in the acidic soil. Next micronutrient is the chlorine. So chlorine has a role in the photosynthetic oxygen evolving complex and a charge balance across the cellular membrane. So these are the two main functions of the chlorine. So uh, chlorine ion is ubiquitous in the nature and highly soluble. Thus is rarely deficient. Means chlorine deficiency is rarely seen in the plant, and deficiency normally can be shown only in the very carefully controlled solution culture experiment. Means in natural conditions you never see the deficiency of chlorine in the plant. The chlorine deficiency only seen in a control experiment, in a control solution culture experiment. Along with the manganese, so again as we know that manganese also involved in the oxygen evolving complex functioning, chlorine is also required for the proper functioning of these oxygen evolving complex. Next, chlorine is a highly mobile anion and with two principal functions. One is the it maintain the electrical neutrality across the membrane, and another one is the it function as a osmotically active solute in the plant vacuole. So these are the two principal functions of the chloride ion. So chloride ion appear to be required for the cell division in the both leaves and shoots. Chloride ion is readily taken up, and most plant accumulate chloride ion for far excess in their tissue excess in their minimal requirements. Means apart from minimal requirement, the chloride ion absorbed by the plant and accumulate in the plant tissue. Plant showing deficiency of the chloride exhibit the different kind of symptoms, and one is the again uh, growth is reduced, wilting of the leaf tips, and a normal chlorosis or general chlorosis. So these are the some deficiency symptoms of the chloride ions. Next one is the nickel. So nickel, the functions of the or role of the nickel is not clear. So nickel is recently added in the list of essential nutrients. Nickel is abundant in the metallic elements and readily absorbed by the roots. In ubiquitous plant tissue, the range of the nickel normally 0.05 to 5 mg per kg of the dry weight. One of the principal difficulty encountered to attempt to establish a role of nickel is the extremely low requirement. Means this micronutrient is required in extremely low concentration by the plant. Means uh, if the concentration is higher, then this showing some kind of toxic effect on the plants. It has been estimated that the quantity of nickel needed by the plant to complete its life cycle is approximately 200 nanogram. So, a requirement that can be met by the initial nickel content in the seed in the most of the cases. So, that amount of nickel already present in the seed. So, plant did not need any extra nickel for their functioning. In order to establish a nickel deficiency, it is necessary to undertake the extensive purification of the nutrient salt and then grow several successive generations to the seed in the nickel deficiency solutions. So, in normal condition, it is very difficult to see the symptoms of nickel deficiency symptoms of the nickel. The strongest evidence in the favor of essential status of the nickel is based on the studies with the legumes and cereal grains. The basics of the nickel requirement by the plant is not clear. Again, it is required in very less amount. So the requirement by the plant of nickel is not clearly, but it may be related to the mobilization of nitrogen during the seed germination. Means if the particular amount of nickel is not present at that time at the time of seed germination, then these seeds do not show proper mobilization of the nitrogen, and seed germination process is disturbed or stopped. So nickel is known as a component of two enzymes. One is the urease and another is the hydrogenase. So urease catalyze the hydrolysis of the urea into the ammonia and CO2 and is found in widely through the plant kingdom. And hydrogenase is responsible for the recovering hydrogen for the use in the nitrogen fixation process. So deficiency of nickel leads to depressed level of hydrogenase activity in the nodules of the soybean, which in turn would be expected to depress the efficient nitrogen fixation. So these are the some important deficiency symptoms of the nickel. This is our last micronutrients. Next is the toxicity of micronutrients. Although micronutrients required by the plants in very less amount or quantity, the micronutrients element are excellent example of dangers of excess. Means if their concentration increase, then they show large toxic effect on the plants. Although they are required in less quantity, less amount, but when they are in excess in the plant system, they show some danger signals or symptoms due to their toxicity or due to their higher level in the plant tissue. Most have rather narrow adequate range. So as we discussed in the first lecture, the adequate range of the macronutrients is large, but in case of micronutrients the adequate range is very narrow. So they become toxic even when the concentration is slightly increased. So become toxic at relatively low concentration, means although the total concentration is low but their adequate range is narrow. That's why that slightly increase in the concentration cause the micronutrients as a toxic. So the critical toxic level defined as the tissue concentration that give rise the 10% reduction in the dry weight. So critical toxic level means a concentration level of any nutrients that cause reductions in the dry weight by 10%. So very widely between the several micronutrients as well as plant species. Next, toxicity symptoms are often difficult to decipher because an excess of one nutrient may induce the deficiency of another micronutrients. For example, the symptoms of manganese toxicity shown in the plants as a brown spot due to the depositions of Mn manganese oxide surrounded by the chlorotic veins in the waterlogged soil. But excess of manganese may also induce the deficiency of iron, magnesium, and calcium. Means manganese compete with the both iron and magnesium for uptake, and manganese binding for the binding to the enzymes. Means if the manganese is higher in concentration in the plant tissue, at that time it inhibits the iron and magnesium for uptake. Level of manganese is higher, at that time it inhibits the calcium translocation in the shoot apex and causing a disorder known as crankle leaf. Thus, the dominant symptoms of manganese toxicity may be actually the symptoms of deficiency symptoms of iron, magnesium, and calcium. So we can say that one micronutrients toxicity result in the deficiency of another micronutrients. So in this example, with the help of this manganese example, we can show that if the toxicity of manganese is increased, means its level is increased, at that time the deficiency symptoms appeared due to the deficiency of iron, magnesium, and calcium, because higher level of manganese compete with the iron and magnesium for uptake and inhibit the calcium translocation. So because of that, plant lack of iron, plant lack of magnesium and calcium, and because their deficiency symptoms are appeared mainly. So we can say that toxicity symptoms are often difficult to decode. Means uh, in case of micronutrients is very hard to decode the toxicity symptoms caused by a particular micronutrients. So this is our last slides, and hope you understand this topic. Try to make a table using all these information spread among the different slides, and thank you for watching. Hope you like this, and I will see you in the next lecture.